Today, first of all, I'd like to thank the Provost and Chuck Tabor, who are not here, as well as committee members who, for first of all, organizing this Provost lecture series, as well as inviting Catherine to come and present her research. So when I was thinking about doing an introduction to Catherine, I was trying to think of a word that might describe her, right? And I'm sure many of you who know Catherine could think of many words, but I came up with the one energy. Energy, because Catherine has this unbridled energy for doing research, for the laboratory, for her family, I've discovered, for the laboratory, and for the graduate programs, but also for Stony Brook University, as you're going to see as we discuss this whole topic. So again, Catherine's an MD PhD. She did her undergraduate work at Swarthmore College. For those unfamiliar with it, she's already finished her first 2 years of medical school, and she is now finishing her 4th year of graduate school and will be re-entering into medical school this summer. So 4 years, as most of us who are PIs know, that's really when graduate students become useful, okay? But Catherine, even in this relatively short time frame, has already published 2 manuscripts, first author on both of them. One of them was also reviewed by Faculty of 1000. And the work she's going to show today, only about half of this is published. Right? So she's, in a 4-year period, she's been very successful in the laboratory, getting lots of work done. She's also introduced into my lab a number of techniques that's allowed my lab to submit a new R01, which will be reviewed this June. Let's hope everything goes okay. But it's really based on Catherine's introducing the techniques as well as much of the research she's done. So in terms of the laboratory, you can see she's been quite energetic. She's also won several awards. She's won an award from the Society for Neuroscience. I don't remember the specific name of it, but perhaps more relevant to me, or those of us who are PIs and have to pay people, she has won a National Research Service Award. So she's had an award that pays not only her salary now, but also is going to pay her salary when she re-enters medical school. And she's also, I think anybody who's the director of the MD PhD program, that would be Mike Froman, or myself when I was the program and neuroscience director, and Joel Levine now, we recognize that Catherine really participates a lot in these programs, right? She's interviewing students, she's organizing journal clubs, she's going out to dinners with candidates, all these things, okay? So you would come to that point and you would say, wow, this is amazing. This person's done all this in 4 years, okay? That's nothing yet, right? So again, come back to this energy, this thing about Catherine's commitment to the university and to the medical program and to the graduate program in neuroscience. So this is a list of the committees she has served on or continues to participate in. Now, I'm not going to go through this whole list. What I will mention is just a few of them. So she's been actively involved in the graduate student organization, the finances, other issues related to that. I can't list all of them there. She's a board member of the Stony Brook Child Care Committee. She's on the advisory committee for the campus recreation. She's on the parking appeals board, so now I know why my ticket request got rejected. She's been on academic judiciary, and she's been on the search committee for the vice president for research. And finally, she's on the Stony Brook Council, which I think most of us are aware of is this sort of umbrella organization that overlooks Stony Brook. It includes the president, the provost, the deans, and other people of great importance. And so I was at the basketball game on Saturday when Stony Brook played Vermont, and I thought, well, you know, I got these really nice seats, I can look out, I've got great view. And of course, looking down there, sitting in front of me, sitting in a better seat, sitting amongst great celebrities, the Gord Jefferson former mayor, the provost, was Catherine, because she had the honor of sitting in the seats associated with President Stanley, okay? So anyway, I hope you see the energy in this, and again, I hope you appreciate the effort and time she has put into this, the research that she's going to present today. Thank you. And here's Catherine.
Okay, thank you. So today I'm going to talk to you about mechanisms of ionotropic glutamate receptor assembly. So in the brain, there are over 10 to the 11th cells, or 100 billion neurons, that are responsible for perceiving all the different environmental stimuli and taking that information and conveying it into an appropriate response. So one of the important features of the neuron is that it is polarized and ensures that information flows in a single direction, beginning at the dendrites down to the axon terminals. And facilitating this flow of information are 4 distinct morphological features of the neuron: the dendritic tree, the cell body or the soma that contains the metabolic machinery of the cell, the axon, and the axon terminals. So incoming stimuli, both excitatory and inhibitory, come in on the dendritic tree, and these different signals result in an electrical potential, the generation of electrical potential that is then transduced through the cell body, and all of these different excitatory and inhibitory signals then are summated or integrated at the level of the cell body. If the summation of these signals reaches a certain threshold, an action potential or an electrical signal will fire. And this action potential is regenerated and it is propagated down along the length of the axon to the axon terminals. Now, this action potential or this electrical signal can be propagated either to other local neurons, so short distances, as well as very far distances, up to 3 meters long. So this electrical signal then reaches the ends of the axon terminals, also known as a presynaptic terminal, and results in the depolarization and the release of a chemical, a neurotransmitter, that can then signal to the next adjacent cell. And so it's really this pattern of electrical to chemical to electrical signaling that's important for nervous system function. And so what I'm going to show you is this animation in which we'll see from this cell body here an action potential that fires and is propagated down along its axon and is going to stimulate a secondary or a postsynaptic neuron. Then it's going to zoom in and we're going to see actually these axon terminals, the depolarization of the presynaptic cell, the release of neurotransmitter across the synaptic cleft, and then the subsequent activation of postsynaptic receptors that generate an electrical signal in the postsynaptic cell. So this is a little fast, so it'll play twice. But we can see the action potential is getting shot down the axon, stimulates that cell soma, and now this presynaptic terminal is going to be depolarized, releasing neurotransmitter across the synaptic cleft and eliciting an electrical signal in the secondary neuron. So it really is this electrical to chemical to electrical signal that's important for nervous system function. And so now I'm just showing a cartoon of this scene of the synapse. And what we can see is, again, this presynaptic action potential is propagated down along the length of the axon, and the depolarization of the presynaptic terminal results in the fusion of vesicles that contain glutamate, or neurotransmitter, the chemical signal, that can then be released into the synaptic cleft. The chemical signal is very important, and it allows for amplification of neural signals within the brain. And as it diffuses down its gradient, it can bind to receptors embedded in the postsynaptic membrane. Glutamatergic signaling, glutamate will bind to glutamate receptors such as the AMPA or the NMDA subtype, and upon ligand binding, the channel opens and there's ions that flow across the membrane. This flux of ions across this postsynaptic membrane generates an electrical signal, also known as an excitatory postsynaptic signal, allowing for this electrical chemical neurotransmission. On the right here is a high-resolution electromicrogram of the synapse, with the synaptic cleft indicated by this asterisk. And you can see that at the presynaptic membrane, there are these vesicles that are tethered near and directly opposed to that of the postsynaptic terminal. And this thickening is the postsynaptic density, which contains the glutamate receptors as well as other auxiliary proteins that facilitate the trafficking and clustering of glutamate receptors at the postsynaptic membrane. And so glutamatergic synapses actually are present at over 70% of the synapses in the brain. And so glutamate signaling is very important to normal nervous system function. And it's also very critical in early neural development, being involved in neuronal migration as well as corticogenesis, and underlies the mechanisms for higher order cognitive functions such as pain perception and learning and memory. Since glutamate is so prevalent in the system, it is not surprising that dysfunctional or aberrant glutamatergic signaling is also implicated in many nervous system diseases. For instance, acute excitotoxic diseases such as epilepsy or stroke are due to the increased activation or hyperfunction of glutamatergic receptors. Meanwhile, schizophrenic diseases are actually thought to occur due to, psychiatric diseases such as schizophrenia and bipolar disorder are thought to occur due to a decreased functioning of receptors. And finally, glutamatergic signaling has also been implicated in neurodegenerative diseases such as Alzheimer's and Parkinson's. So since glutamate is present at over 70% of the synapses, how and is important for both physiological and is involved in pathological processes, what's regulating this system? What's making it so dynamic? So we can think of it from two ways. First, what are some of the presynaptic factors that are mediating glutamatergic signaling? And these include, the major one is the amount of glutamate that's released at the synapse. So this has two factors that can influence this. One, what's the probability that a vesicle will fuse to the presynaptic membrane and release its contents into the synaptic cleft? And two, the amount of glutamate molecules or the quanta that are in the vesicle and released. So if we have increased glutamate release, there's an increased probability that it will be able to bind a receptor and elicit an electrical signal. So now we can also then consider the different postsynaptic factors that can influence synaptic transmission. And there are three main factors. First, the number of the receptors that are located in the postsynaptic cell. So studies have shown that on average there are anywhere from 10 to 100 glutamate receptors that are localized in the postsynaptic density opposite of the active zone or the release sites for glutamate. And if we increase those, the number of receptors, we'll have an increase in the magnitude of the electrical signal that's generated upon glutamate activation. Also, the location of these receptors is also very important. So if we have 10 receptors that are in close proximity to the glutamate release, they are most likely going to be activated. But if you have another 20 receptors that are further away from the site of glutamate release or extrasynaptic, there is a less likelihood that these receptors will see glutamate and be activated. So it is both the number and the location of these receptors in response to glutamate release that's important for determining the magnitude of the postsynaptic potential. Finally, gating properties also contribute to the shape of the postsynaptic potential. So the two major types of glutamate receptors that are present in the postsynaptic density are the AMPA and NMDA subtypes, and they have different kinetic properties. And so if we compare their responses or their current amplitudes to that following glutamate application, we see that AMPA receptors have a very fast onset or turning on of the channel, and then upon glutamate removal also turn off very quickly. Whereas NMDA receptors turn on quickly but take a much slower time course to reach the peak current amplitudes. And then upon removal of the agonist also turn off slower. So it is a combination of both the number, the location, and these different gating properties that determine the magnitude and the shape of the excitatory postsynaptic potential. And today what I'm going to focus on is specifically with regards to the number. So how does changing the number or the amount of receptors expressed on the surface influence glutamatergic signaling? So glutamate receptors are tetramers formed by 4 subunits. And recently, about 2 years ago now, the first crystal structure of a glutamate receptor was crystallized of an AMPA receptor. And what the crystal structure allows us to see is that glutamate receptors are highly modular. They have an extracellular amino-terminal domain, an extracellular ligand binding domain that is able to bind the agonist, the transmembrane domain, which contains the ion channel, and not pictured here is a large C-terminal domain. So, just to refresh, the amino-terminal domain, as well as now we have the ligand binding domain, which is formed of 2 discontinuous polypeptides, the S1 and the S2, that come together to form a clamshell that can then close upon ligand binding. The transmembrane domain is formed by 3 full-pass transmembrane segments, M1, M3, and M4, and a reentrant port loop, M2. And what's going to be really important to this talk is that the ion channel core is formed by the M1 to M3 segments. And so this is what's really lining the ion channel. 4 of these such subunits come together to form a functional tetrameric receptor. And in the brain, the majority of glutamate receptors are actually heteromers. That means they're formed by 2 similar subunits. So I've just indicated this here with a slightly different color shading. So glutamate receptors share a similar homology to that of other types of glutamate other receptors, such as the 2 transmembrane potassium channels. And it's really this ion channel core that is homologous to this, albeit it's inverted in the membrane. What's important is that these 2 transmembrane potassium channels are functional, and also that this ion channel core is also similar to that of a prokaryotic ancestor, prokaryotic glutamate receptor, GluR0. What separates eukaryotic glutamate receptors from that of the 2 transmembrane potassium channels and GluR0 is this additional transmembrane segment, the M4 segment, that is C-terminal to the ion channel core. By also having this M4 transmembrane segment, it places the highly regulated C-terminal domain, which is important for trafficking and clustering of receptors at the synapse intracellularly. So, what is the functional significance of this M4 transmembrane segment? So to begin, we first wanted to look at GluR0 and see how, and we express this construct, which is homologous to mammalian glutamate receptors with regards to the ion channel core, and we express this in HEC293 cells and apply glutamate. And we see that indeed the channel, these constructs gate, so they must be able to form tetramers and be expressed at the membrane. But what about mammalian glutamate receptors? So what we did is we wanted to understand what's the role of this additional M4 segment in mammalian glutamate receptors. So to do that, we introduced a stop codon in the wild type GluA1 to make a construct known as the delta M4 construct that is topographically homologous to that of GluR0. We then took these constructs and expressed them in Xenopus oocytes and applied glutamate to look at their glutamate activated currents. And for wild type, when we apply glutamate, we see activation of the channel, inward currents, and glutamate activated currents that are consistent upon repeated applications. Well, what happens now if we apply glutamate for our delta M4 construct? When we apply glutamate for delta M4, we see the lack of any functional glutamate activated currents. So this would suggest that the deletion of the M4 segment reduces functionality. But what actually is accounting for this delta M4 phenotype? There are possibly 3 possible interpretations of this result. First, it could be that the delta M4 construct is resulting in a gating defect. That is, that these receptors are able to form tetramers and they're expressed in the membrane, but when glutamate binds, the channel cannot open. So it cannot pass any ion, allow any ions to pass and generate these current amplitudes. Second, if you remember, when we remove this delta M4 when we remove the M4 segment, we also remove this highly regulated C-terminal domain. So perhaps this loss of functional occurrence is actually due to deletion of the C-terminal domain, which others have shown is important for trafficking and anchoring of the receptors at the synapse. Finally, the third interpretation could be that perhaps this delta M4 construct is causing a deficit in the assembly or the biogenesis of these receptors. So to first determine whether or not the C-terminal domain is responsible for this phenotype, we generated a second construct in which we now left the M4 segment intact, but introduced the stop codon, just removing the C-terminal domain. And when we expressed the construct in Xenopus oocytes and applied glutamate, we saw that, like wild type, we get rapid activation of the channel, inward currents, and current amplitudes that are being able to be repeated with repetitive glutamate application. And if we quantitate the current amplitudes for our delta CTD construct and compare that with GLU-A1, we see that the magnitude of the current amplitudes are very similar to that of wild type, suggestive that the delta M4 phenotype is not due to the absence of the C-terminal domain. So what about the role of biogenesis? Is the M4 transmembrane segment necessary for glutamate receptor assembly? To answer this question, we looked at surface expression of these receptors in HEC293 cells using immunocytochemistry under non-permeabilized conditions. So when I took HEC293 cells and co-transfected with GFP and either wild type or the delta M4 construct that contain an HA tag, and then stained with an anti-HA antibody, for wild type, we see abundant surface expression. However, when we look at the non-permeabilized delta M4 constructs, we don't see any detectable surface expression, suggesting that the M4 segment is necessary for glutamate receptor assembly. But this delta M4 construct or mutation is a dramatic mutation. You're removing an entire transmembrane segment. So what we wanted to do was now see what would happen if we introduce single point mutations throughout the M4 segment and how this would affect surface expression and functioning of the channel. So what we did is we introduced tryptophan residue, which is a big bulky amino acid that disrupts protein-to-protein interactions throughout the M4 segment. And then we expressed these new tryptophan substituted mutants in Xenopus oocytes and looked at their glutamate activated currents. And we saw one of three phenotypes. We saw those constructs that generated glutamate activated currents similar to that of wild type, those that showed reduced currents, and those as pictured here at G798 that showed a loss of function or no detectable currents. And so we took this data and we then quantitated it and put it in this graph so you could see that there are those positions that show normal functioning glutamate activated currents, decreased currents shown in blue, and then the positions marked with an X were non-functional. And so these non-functional or non-detectable currents are also highlighted over here in red. And this is to familiarize yourself because you'll be seeing this a lot throughout the rest of the talk. So we know that these single tryptophan mutants can replicate the delta M4 phenotype at least from a functional or electrophysiological standpoint, but what about with regards to assembly? So again, we used immunocytochemistry to look at the surface expression now of these single tryptophan mutants. And shown here are these positions marked with an X, which were non-functional, that did not show any functional glutamate activated currents, expressed in HEC293 cells. And when we stain for an antibody specific to the N-terminal domain of GLU-A1 and look at the surface expression, for each of these positions, there was no apparent surface expression, no detectable surface expression. However, if we look in permeabilized conditions, when we can look at intracellular stores of proteins, we see that the protein is indeed being synthesized. So this would support the idea that the M4 transmembrane segment is necessary for surface expression. And so if we take this data from both our tryptophan mutagenesis screen as well as the immunocytochemistry and map this onto a helical net, which is a two-dimensional representation of an alpha helix, we can see that these positions that show no surface expression shown in red fall along a single face of the alpha helix. And we actually can take this data and map it onto the crystal structure for the M4 segment. And it just nicely points out that there's this unique face of the M4 segment that's necessary for surface expression. And further, when you now take this face and you model it in the crystal structure, you see that this face aligns closely with the transmembrane segments of an adjacent subunit, suggesting that the interaction of this M4 segment with the M3 and M1 transmembrane segments is necessary for surface expression. So far, just to recap what I've shown you, is that the M4 transmembrane segment is required for surface expression, and either deletion or single tryptophan substitution results in the loss of detectable surface expression. Further, the delta M4 phenotype is not a result of the C-terminal domain, but rather due to the intrinsic nature of the M4 segment itself. And finally, based on predictions from the crystal structure, we believe that it's the interaction of the M4 segment with the adjacent transmembrane domains that's necessary for surface expression. So to begin to get at mechanisms of assembly, we first have to take a step back and understand how glutamate receptors are formed, or the biogenesis of glutamate receptors. And so glutamate receptors are functionally tetramers, but they are formed from monomeric subunits that first form a dimer, and then two dimers come together to form a dimer or dimers, or a tetramer. And this occurs largely in the endoplasmic reticulum. So once proteins are synthesized, they're translocated to the endoplasmic reticulum, where with the aid of chaperones can then fold and assemble and then finally begin to form dimers. These two dimers will then come together and form a tetramer. And it's only when the receptor is in its tetrameric form and is deemed competent that it can be exported from the endoplasmic reticulum. Once released from the endoplasmic reticulum, it receives further post-translational modifications in the Golgi body before being inserted into vesicles that are then expressed at the plasma membrane. And so, to be able to understand this process, we also want to be able to look at the native oligomeric state of these proteins. And to do so, we have utilized a technique known as blue native page, which allows us to look at the oligomeric state of proteins under non-reducing and non-denaturing conditions. So if we take a wild-type AMPA receptor and purify the protein and run it on a blue native page gel, we will see the prominent presence of tetramers as well as dimers. And it's this tetramer pool that represents the receptors that are expressed on the plasma membrane and that are able to generate glutamate-activated currents. This dimer pool represents receptors that are pools of dimers that are present in the endoplasmic reticulum. And other labs have shown is that these are very important for the dynamic regulation of glutamatergic signaling, that there's ready pools of receptors that can be trafficked to the membrane upon stimulation. So the crystal structure is very modular, or the glutamate receptor structure is very modular. It's also very important for glutamate receptor biogenesis. Studies have shown that the amino-terminal domain is important in the initial formation of dimers, and that both the ligand binding domain and the transmembrane domains have a role in ensuring the competence of these receptors and aiding in their export from the endoplasmic reticulum. However, the transition from dimers to tetramers is not well understood. And this is something we're very interested in. So, as I alluded to earlier, based on predictions from the crystal structure, we believe it's really this interaction of the M4 segment with the adjacent transmembrane or the ion channel core that is critical to glutamate receptor assembly. And so, to look at this, just to remind you that we want to understand this. How is the M4 transmembrane segment involved in receptor assembly? And so if we delete the M4, we lose surface expression. So to get at this question, we now want to see where is this delta M4 construct being retained? And to answer this, we use immunocytochemistry now under permeabilized conditions to look at the intracellular distribution of our delta M4 construct. And by using antibodies specific for either the endoplasmic reticulum, calreticulin, or the Golgi, GM130, we can look at the co-localization of our protein with these different markers and have an idea of where this protein is being retained. So if we look at the staining, we stain for our HA tag, as well as either the ER marker or the Golgi marker, we see that our delta M4 construct is highly co-localized in the endoplasmic reticulum. And so the key steps for export from the endoplasmic reticulum is to be in a tetrameric form. So our next question is our delta M4 construct able to form a tetrameric receptor? And so we utilize Blue Native Page to ask this question. And again, for wild type receptors, we see prominent dimer, tetramer and dimer formation. And when we analyze our delta M4 construct, we now only see dimers. So this would suggest that the delta M4 construct is incapable of mediating this transition from dimer to tetramer. But this mutation, the delta M4, is such a dramatic mutation, and it also limits our ability to ask more interesting questions to really get at the mechanism of this transition from a dimer to a tetramer. So what we want to do is go back to our single tryptophan mutants. And so we wanted to look at the role of this phase, this non-functional phase, that's important for surface expression, and see if a single point mutation can also disrupt tetramerization. And so if we look at either wild type or a functional mutant, so L811, which gave us functional currents when we introduced this tryptophan, we see that there are both dimers and tetramers. But what happens if we now look at a small subset of our red positions, or our non-expressing positions that fall along the face of the M4 segment, glycine 802 and glutamate E813, and we look at their ability to form tetramers. So when we run these samples on a Blue Native Page, we see that for our non-functional red positions, we only see dimer formation. So this would suggest that indeed the M4 segment, this specific face of the M4 segment, is necessary for tetramerization. However, you might question, well, how can you tell that this protein is just not being misfolded and being targeted for degradation? So we have also looked at the oligomeric state of these proteins using a second technique. So in collaboration with Hiro Furukawa at Cold Spring Harbor, we're utilizing fluorescence detection size exclusion chromatography to look at the native oligomeric state of these proteins. And one thing that FSEC allows us is to see whether or not the protein is grossly misfolded. So if the protein was misfolded, we would see a peak early on in the chromatogram. And so what I'm showing here now is the chromatogram profile for wild type GLUA1. And we see a prominent tetramer peak and a dimer. So now, what happens when we introduce single point mutations? And you can see for a small subset of these red positions that when we alter the interaction by introducing a tryptophan or even an alanine, we disrupt the chromatogram, the profile, and we have little to no tetramer formation, but abundant dimer formation. So this would suggest, this is consistent with the notion that the M4 segment is required for tetramerization. We can also look at this for GLUA2. So for a second AMPA subunit, is this consistent for all AMPA receptor subunits? And for wild type, again, GLUA2, we see a prominent tetramer peak, and perhaps some indiscriminate dimers or monomers. And if we now, again, look at that functional leucine L811 tryptophan, again, we know that it's on the surface, at least by Blue Native Page, it forms tetramers, and here, this is confirmed with our FSEC results that, indeed, we're forming functional tetrameric receptors. So what about our tryptophan mutants that disrupt surface expression? When we look at them on Blue Native page, again, we see that there's little to no tetramer formation, but very prominent dimer formation. Consistent with the hypothesis that M4 mediates tetramerization, but does not play a large role in the dimer formation. So our tentative model is that in the endoplasmic reticulum, we have dimer pools, and that these dimers can come together to form a prototetramer that are energetically unstable. And a stable tetramer is only formed when the M4 segments interact with the adjacent ion channel core. And so we really need this wrapping around and association of the M4 segment to stabilize this tetramer. And any disruption of this interface of the M4 segment with the ion channel core, the M1 and the M3 at the adjacent subunit, will prevent tetramerization. So based on that model, we should then be able to substitute positions in the M3 segment and also disrupt tetramerization. And so what I show here on the left is our M4 face, and based on predictions from the crystal structure, interacting partners in the M3 segment within a 5 angstrom radius. And so what we wanted to do now is mutate specific positions in the M3 segment and see whether this disrupts tetramerization. And so first we looked at the surface expression of these receptors. And the positions that are shown on the right are bolded here in the chart. And we can see that for wild-type receptors, we see abundant surface expression. But now when we introduce a leucine residue, which is not as dramatic as a tryptophan residue, but should still interrupt protein-protein interactions, we see that, that there is little to no surface expression. And one of the key things, though, to note is that although we see some surface expression for this one particular instance, this one mutation, this is not a one-to-one interaction. So there might be multiple interacting partners that are important for this membrane phase. So perhaps if we mutate one other receptor, we might lose all surface expression here. So we wanted to now look at these profiles using FSEC, fluorescent size exclusion chromatography. And again, the red trace is wild type, showing a prominent tetramer peak. And when we look at our leucine mutations, we see little to no tetramer formation and prominent dimer formation, suggesting that indeed mutations of residues of interacting partners in the M3 segment do alter tetramerization. So to remind you, it's this model that we really need the M4 segment to wrap around and stabilize the formation of the tetramer. So we wanted to ask, well, can this process of M4-mediated tetramerization be regulated? And so this is some preliminary data that we have in the lab that begins to get to ask the question, can M4-mediated tetramerization be regulated? And what we're doing is altering the size of the side chains at a specific position, V valine 795, to either smaller or larger residues. And so if we look at our wild-type trace, when there's a valine, we know that we see prominent tetramer formation. And if we mutate this valine to the tryptophan, we know that there's no functional occurrence, no surface expression, and by both Blue Native page and FSEC, we only see dimer formation. So what would happen if we take this valine and now mutate it to a smaller amino acid, alanine, or to a larger amino acid, leucine? And so if we look at this via FSEC, we see that in comparison to wild type, by changing the valine and mutating it to a smaller residue alanine, there's an increase in the apparent rate of tetramer formation. And further, if we now look at the chromatogram profile for the valine to leucine mutation, we see that although we are still forming tetramers, there's an increase in the amount of dimer formation, suggesting that this increase in side chain size is preventing the rate of tetramerization. So it would suggest that there's an inverse relationship of size to the rate of tetramerization, and that these interactions are very important. We can also look at this functionally, and this data was collected by another graduate student in the lab, and what we've shown is that both the functional data supports what we're seeing from our FSEC, in that if we look at glutamate activated currents for wild type and compare that with our valine to alanine mutation, we see an increase in current amplitudes, which suggests that there's an increase in tetramer formation and an increased expression of these tetramers at the cell surface. Whereas when we look at the glutamate activated currents for our valine to leucine mutation, there's a decrease in current amplitude. And we can actually graph this, the current amplitudes versus the size of the side chain, and see that there is an inverse relationship between the size of the side chain with the amount of current amplitude. So this would suggest that, yes, perhaps we may be able to regulate the formation M4-mediated tetramerization by changing the interactions of the M4 segment with the adjacent transmembrane domains. And so what I've shown you today is that the eukaryotic-specific M4 transmembrane segment mediates tetramerization, and that either deletion of the M4 segment or single tryptophan substitutions can disrupt tetramerization but do not affect dimerization. Further, we have begun to show some preliminary evidence that this role of the M4 segment in tetramerization may be regulated. And also, future studies we're interested in studying is, since the M4 segment is directly tethered to the highly regulated C-terminal domain, and which is important for trafficking, whether this also may play a role and regulate M4-mediated tetramerization. And finally, this role of the M4 segment in receptor oligomerization may represent a therapeutic target for drug development to treat glutamatergic disorders. And so I'd just like to thank everyone who's helped me with this work. This work was started by a previous graduate student in the lab, Alexandra Corrales, and of course our Janet, our lab manager, who has made all of our constructs, has helped me out with the immunocytochemistry experiments, has been very pivotal to this project. Other contributors to this work, Jean, Rishek, and Ihab. Our collaborators, Pooja, Hiro Furukawa, and Mark Bowen. My thesis committee members, and my funding sources. Thank you.